Okay, so then the final talk of our session is by Vilmante Borutaite. Sorry if I got that wrong. And she's going to talk about extracellular tau-induced neuroinflammation and neuronal loss.
Thank you. First of all, I would like to thank Guy and team for inviting me and giving a chance to present our work, how extracellular tau interacts with microglia, causing neuronal death. I'll start with a well-known medical statement that it is important to have a good sleep, and especially around midnight. Various reasons can be given explaining why it is important, but one relates with recently found glymphatic system, which functions in the brain, circulates along brain arteries, and which is most active during midnight. And function of this glymphatic system is to remove metabolic waste products, excess of glutamate, and also to remove pathogenic proteins such as beta amyloid and tau proteins. The fact that glymphatic system removes tau proteins every night and removes these proteins from healthy brain seemed for us intriguing, because tau is intracellular protein and it shouldn't be outside the cells. So tau is mainly cytosolic microtubule-associated protein. 6 isoforms have been identified in adult brains, and they differ in repeat areas in C-terminal and N-terminal. Function of all these isoforms is to maintain microtubule stability, and it is known that in pathology tau can be phosphorylated, and it can then aggregate and form intraneuronal fibrillar tangles thought to be involved in neuronal dysfunction and death. However, recent studies have shown not only that tau is flushed out with the glymphatic system, but studies have shown that tau can be secreted from neurons, and particularly from actively functioning neurons in exosomes. However, the mechanism of secretion nor functions of this extracellular tau protein are not known. And this prompted us to investigate whether extracellular tau can be neurotoxic. In our experiments, we used primary mixed neuronal glial co-cultures isolated from rat cerebella, and we treated these cultures with various tau proteins. So the longest isoform, 2N4R, slightly shorter, 1N4R, and short C-terminal peptide. And we found that no one of these proteins gets any effect on neuronal viability over 48 hours of incubation. But slightly different situation was with phosphorylated form of 2N4R protein. This protein, phosphoprotein, at low 30 nanomolar concentrations had no effect on neuronal viability. However, at higher concentrations, 700 nanomolar concentrations, it caused a neuronal death, which was mainly necrotic, as can be seen in these photos, where red color indicates necrotic nuclei. At this concentration, phospho-tau killed not only neurons, but it killed also microglial cells. In contrast, lower concentrations of phospho-tau, 30 nanomolar concentrations, stimulated microglial proliferation, And at this concentration, phospho-tau was found to reduce neuronal numbers in cell cultures, indicating that phospho-tau is most toxic among tau proteins investigated, causing loss of viable neurons at low concentrations and necrosis of neurons and microglia at highest but still submicromolar concentrations. The longest isoform 2N4R also caused proliferation of microglial cells and also stimulated proliferation of astrocytes, but to a lesser extent. And in these experiments, we tested not only monomeric tau protein, but also various aggregate forms, small oligomers and large oligomers, which were prepared during 24-hour incubation at room temperature or in the presence of beta amyloid. But all these oligomers, as well as monomers, stimulated to a very similar extent proliferation of microglia. All forms, aggregate forms of tau protein were not toxic to neurons. They did not reduce viability of neuronal cells over 48 hours incubation. However, the number of neurons in cultures was found to be reduced when cell cultures were exposed to 3 micromolar extracellular tau, and this loss of neurons was already observed after 24 hours, and the number decreased further during 48-hour incubation, showing that tau causes neuronal loss from CGC cultures independently of aggregation state. So, do we see here phagoptosis? It is neuronal death by phagocytosis that Kai presented earlier today. And to elucidate this, we investigated whether microglial cells are involved in extracellular tau-induced removal of neurons. In our previous experiments, we used mixed neuronal glial cultures in which microglia, there were 6% of microglia. And when such cultures were pretreated with LME, it's lysyl methyl ester, which selectively removes microglia, you can see here that cultures were practically without microglia. And when such cultures were exposed to extracellular tau, tau was not able to induce neuronal loss in these cultures, indicating that tau is not toxic to neurons in the absence of microglia. Further, we investigated whether tau can stimulate microglial phagocytic activity. Here we used microglial monocultures, which were incubated with tau for 24 hours, and then fluorescently labeled latex beads were added to the cultures. And we found that after such treatment with extracellular tau, microglial cells phagocytosed in average 4 times more beads than control cultures, indicating that extracellular tau stimulates phagocytic activity of microglia. So these experiments show that tau affects microglia directly, but what about neurons? Are they directly affected by tau protein? And do tau-affected neurons expose eat-me signal, which is phosphatidylserine on cell surface? And indeed, we found that incubation of CGC cultures with tau resulted in exposure of phosphatidylserine, which was detected by annexin labeling, and exposure of phosphatidylserine was observed already after 6 hours and increased during 48-hour incubation. And interestingly, if these CGC cultures were first pre-treated with RSC, which prevents proliferation of glial cells and therefore removing microglial cells, in such conditions, tau did not cause exposure of phosphatidylserine on neuronal cell surfaces, again suggesting that tau causes exposure of phosphatidylserine on neuronal cells not directly, but the activation of glial cells. Next question was, what is the mechanism by which extracellular tau stimulates microglia to affect neurons? Here we were interested whether this mechanism involves activation of toll-like receptor 4, this receptor mediated inflammasome activation, because it was known that some pathogenic aggregated proteins like beta amyloid, they act on microglial cells through activation of this receptor. And in these experiments, we used antibodies against toll-like receptor 4. When cell cultures, CGC cultures, were pre-incubated with this antibody, this had no effect on extracellular tau-stimulated microglial proliferation, indicating that microglial proliferation is probably independent of activation of this receptor. However, antibodies against toll-like receptor 4 reduced microglial phagocytic activity stimulated by extracellular tau, and also these antibodies prevented extracellular tau-induced neuronal loss, indicating that antibodies against toll-like receptor prevent tau-induced phagocytosis of neurons, but not proliferation of microglia. Activation of toll-like receptors usually leads to inflammasome formation and activation of caspase-1. And we were interested whether caspase-1 is also activated by extracellular tau, and particularly in which cells caspase-1 can be activated by extracellular tau. In these experiments, cell cultures were exposed to tau for 24 hours, and after that, cell cultures were stained with Famflicker, which detects active caspase. And indeed, we found that after this 24 hours incubation with extracellular tau, Famflicker signal increased in these cultures. And the signal coincided with microglial cells, which were stained by a specific indicator, IBA4 for microglia, indicating that caspase-1 is activated by extracellular tau in microglial cells. And when we treated the cultures with toll-like receptor 4 antibodies, this caspase activation was reduced, showing that tau, extracellular tau, induces activation of caspase-1 in microglia via activation of toll-like receptor 4. Inhibition of caspase-1 by selective inhibitor of this caspase, reduced extracellular tau-induced phosphatidylserine exposure on neuronal cells, and also inhibitors of caspase-1 prevented extracellular tau-induced neuronal loss from cell cultures. Now, activation of inflammasome and caspase-1 usually results in production of cytokines such as interleukin-1 beta and interleukin-18. However, nor these cytokines nor nitric oxide or TNF-alpha were not produced in tau-treated cell cultures. Nevertheless, exposure of microglial monocultures to extracellular tau resulted in increased production of reactive oxygen species. That was measured using complex red staining, which detects hydrogen peroxide specifically. And pretreatment of microglial monocultures with anti-TLR antibodies prevented this tau-induced increase in ROS production. And incubation of CGC cultures with inhibitor of NADPH oxidase, which may be one of the main sources of reactive oxygen species in microglial cells. So this inhibitor prevented extracellular tau-induced neuronal loss, indicating that extracellular tau stimulates ROS production by microglia via toll-like receptor 4 dependent mechanism, possibly involving activation of NADPH oxidase. And in conclusion, our data let us suggest that extracellular tau-2N4R directly interacts with microglia cells, causing activation of toll-like receptor 4, resulting in activation of caspase-1, increased phagocytic activity, and activation of NADPH oxidase, which then produces hydrogen peroxide, which then can affect neurons, causing exposure of phosphatidylserine on neuronal cell surface, and this results in microglial phagocytosis of stressed but viable neurons. Phosphorylated tau at low nanomolar concentrations possibly acts via a similar mechanism as tau-2N4R, causing microglial phagocytosis of neurons, and at higher concentration, phosphorylated tau causes necrosis of neurons and microglial cells. And finally, many thanks for those people who were involved in this study. So most of the work was done by Katrina Pampuščenko, who is present here, and also Ramunė Morkūnienė. Vytautas Mirnovas and Tomas Schneideris provided us with recombinant tau proteins. Rima Budvytytė and Gintaras Valinčius from Vilnius University. They characterized tau aggregates, and we have long-lasting collaboration with Guy Brown. Thank you.
Thank you. So, any questions? Okay. Sorry. Oh, sorry, back there. I was about to ask one.
Hiya. Oh, sorry. So I'm wondering, what is the source of your tau, and whether you have checked the endotoxin levels or removed the endotoxins, because you have a lot of TLR4 sort of dependent responses.
Yeah. We used recombinant tau protein produced in E. coli, and we always checked endotoxin levels, and we also compared effects of our tau preparations with endotoxins that can also induce some effects similar like here, but we found that at such concentrations that can be present in our tau preparations, LPS had no effect on neuronal loss or nitric oxide production or various other things. So we don't think that it's related to LPS.
Any other questions? I just wanted to ask, you looked at the activation of TLR4. What about TLR2? Is that involved?
We haven't tested this.
And what about ASC spec formation? Because if you're saying you've got activation of the inflammasome, possibly activation of the inflammasome, did you look at that?
We used the inhibitor of inflammasome, and it prevented extracellular tau-induced neuronal loss.
It's quite interesting that you got that caspase-1, because years ago we looked at a peptide called chromogranin A that's in A-beta plaques, and we found it activated caspase-1. And we also didn't see IL-1 beta secretion, but if you inhibited the caspase-1 on the microglia, you didn't kill the neurons. Similar sort of pathway. Mm-hmm. The other thing is, you said at the beginning that, I mean, we know this from people's work at the Institute as well, that tau can be released from neurons in exosomes. Did you ever check the level of your tau in the cultures?
No, we haven't. So because we added extracellularly. So
it'd be quite interesting to make cultures out of neurons that are going to produce phosphorylated tau.
We haven't tested.
Okay, thank you very much. Thanks everybody that spoke this afternoon, including me.